within our suite, we understand that, even aside from classic antibodies or classic biologics formats, we're going away from that, and we have tools which can cover many of these different aspects. We have also tools that can support novel engineering strategies, and many of those are here mentioned on the screen. And we have papers which go into how we've developed those methods and how they were validated. In addition to that, as some of you may be aware, we do have our collaborative enterprise informatics platform, LiveDesign, which now supports natively biologics formats, so antibodies, peptides, and so on.
So Protein FEP works by gradually morphing the side chain from the wild type to mutant through a number of intermediate states along the aerochemical pathway to calculate the phase difference between the wild type and mutant. The FEP+ method was first developed for small molecule protein binding affinity equation, and the slight enhancement of method can also be used to model important endpoints for biologics. So we have extensively validated Protein FEP for thermal stability and protein-protein binding affinity equation. So for protein thermal stability, a very large data set showing on the right part of the panel. You can see that a very large data set with experimental thermal stability measurement on more than 300 point mutations. The FE+ predicted thermal stability result is in very good agreement with experiment, as shown in the middle panel. If we focus on the subset of mutations where the experimental measurement is at neutral pH, as shown on the left panel, the agreement is even better. Similarly, for protein-protein binding affinity, to assess the accuracy of this very important application, we have collected a large number of systems, including 12 protein-protein binding interface, with reliable experimental binding affinity data on more than 200 mutations. The dataset covers small peptide, big antibodies, and other protein-protein binding interface. Here are the results. As you can see on the red plot, the overall RMC between FEP prediction versus experiment is 1.35 kcal/mol. Slightly noisier than the accuracy for small molecule binding, but still very predictive, with a very high level of classification accuracy, and also only less than 10% of mutations has error bigger than 2 kcal/mol. Recently, we have worked out a novel method called FEP+ Residue scanning that enables multiple mutations on multiple sites to be simultaneously modeled in one simulation, offering about 20 times speed up as compared to conventional protein FEP without compromising the accuracy. We have extensively validated this new method, FEP residue scanning, on many, many different systems. For example, on these 12 protein-protein benchmark systems. So we run FEP residue scanning on each case, and the results are almost identical as the benchmark protein FEP, and also in very good agreement with experiment, providing much higher throughput. We have also extensively validated this method on many, many other systems, both in the context of protein thermal stability and for protein-protein binding affinity.
And what we've developed is a hierarchical computational driven approach, bringing FEP residue scanning and protein FEP in order to shorten the project timelines and the cost in order to recover those positive or those important mutations that we are interested in. If you were to compare this with, let's say, a brute force experimental approach, where you experimentally mutate those 39 sites to all amino acids except cysteine, as our collaborators did, making those, testing those, and expressing those, etc. And then you would have 702 mutations. Now, I'm applying this analogy for context. So if you were to take those 702 mutations, you would need 8 96-well plates. And because we have 33 actives, we have around a 4.7% chance of finding a good mutation in any one of those wells. The benefit, though, is that all of those interesting mutations are in that panel. But let's see how we could augment this workflow with computational methods. So if we start with FEP residue scanning, and we look at, well, in this case, not 702, but we're looking actually at a smaller amount. That's because in this case, we've excluded both cysteine and proline. This is something that we'll actually bring online later in the year or in a future release. But for the moment, prolines are still under development. But again, this large library of mutations. Now, if you have access to cloud resources, and you can say, for example, Google, Azure, etc., you can run these on 40 GPUs, and it takes around 5 days. So again, we've already accelerated the timeline already. So we have an idea of our landscape, which mutations are good, which ones are bad. But then we want to take those ones that are interesting, those that are ranked higher, and get more quantitative results. And if we take forward the top 250, let's say, that we again deploy this in the cloud, or if we have our own computing infrastructure, this can take around 2.5 weeks. And this is cost competitive with experiment. Now, what happens then if we don't take our 8 96-well plates, but rather we take 1 96-well plates? What do we get value-wise there? 21 of those 33 mutations are within that panel. It means that instead of having a 4.7% chance of finding a good mutation, we now have a 22% chance, so almost 5 times better. than what we had before. And 64% of all of our good mutations that are possible are within this panel. So we have enriched our experimental testing. We have a better chance of getting good results and good molecules. And in summary, we can get 64% of all of our active hits in a few weeks with 8 times fewer experiments. One of the things I want to mention is that for the modeling services, this workflow that we've mentioned is available as a sort of standard modeling service. Now, one thing that I didn't mention during this approach is that we also use MMGBSA, which is a quicker but much less accurate technique, as another pre-filter where we identify hotspot residues. So identify sites where we definitely shouldn't make mutations because, of course, there's a native salt bridge or a natively strong interaction, and identify and narrow the search space further, followed by FEP residue scan, FEP. We can also apply, once we get to the sharp end of the design funnel, so to speak, we can also combine those top scoring mutations and bring that forward towards lab testing. And of course, just to mention and reiterate, this type of workflow can be applied to affinity engineering, selectivity engineering, cross-reactivity, pH-dependent binding, stability engineering as well. In addition, what we also do as a due diligence, because I did say this is a multi-parameter approach, we can also apply developability parameters, so checking aggregation, checking post-translational modifications, so that again we can shortlist the variants to the most effective and those which are going to be efficacious, which are going to be safe and developable as well.